Hello everyone, and welcome to today's lecture on new research in immunology. So throughout this course, you have learned a lot about various different types of immune processes and that have given you a really solid foundation for understanding how the immune response works. So when thinking about the future of immunology, you can tell by everything that you've learned that every single lecture that you've listened to has an entire future surrounding it. And so this course has really provided you with the groundwork to become scientists, researchers, and clinicians in the field of immunology. Now, thinking about different topics in the future of immunology can be a little daunting, as there are so many different directions in which immunology will be going. However, for today's lecture, we will be focusing mainly on two different topics, namely nanobodies and utilizing viruses in clinical settings, as a framework for you to think about how immunology may look in the future. However, this does not mean that these are the two topics that the future of immunology is limited to in any way. Now, today we'll be talking about nanobodies to begin with. So before we get into nanobodies and what nanobodies are, I want to do a brief review of antibody structures. So antibodies are secreted molecules that come from B cells, and each individual B cell has a highly unique antibody that recognizes a highly specific epitope on an antigen. And if you look a little bit more closely at how antibodies look, they have a variable region up here, and they have a C-terminal region, that's its constant region, down here. And the variable region is made up of a heavy chain and a light chain. And together, the heavy chain and the light chain will recognize the specific epitope of an antigen, such that each unique antibody can recognize a different epitope, which gives our immune systems a very, very high variety of different proteins and other macromolecules that it could recognize, allowing our adaptive immune system to be highly tailored to a variety of different infections that we may encounter. However, antibodies in a large variety of mammals may look like this. However, there are highly unique antibody structures that have been discovered in recent years. Namely, we have our camelid IgGs and our shark IGNARs. So the very unique thing about camelids, which groups llamas and alpacas and camels, and shark antibodies, is that their subportion of antibodies only have heavy chains. So you can see the comparison in this chart here from your textbook, where human IgG molecules have the heavy and light chain. However, camelid IgGs and shark IgNARs only consist of the heavy chain. They're highly devoid of light chains. Really curiously, camelids and sharks evolved this highly independently. So what's really unique about these types of heavy chain antibodies is that first, their discovery came as a very big shock, because you can imagine if you are well versed in classical immunology, you understand that antibodies are made of two chains. So you can imagine the surprise that researchers had when they discovered antibodies in different animals that only had a heavy chain. For a long time, this was an area of confusion, but in many years of research, we have come to discover that these are true antibodies that work highly efficiently. The other curious thing about these so-called heavy chain antibodies is that if you look at their Fab domains of these heavy chain antibodies, they become single domains. So if you take a human IgG immunoglobulin and you just take its Fab portion seen here, it still has the two chains. It still has a heavy chain portion and a light chain portion that is made up, again, of a variable region and a constant region. And these are pretty bulky molecules. They're about 50 kD. However, if you look at immunoglobulins that are heavy chain antibodies, seen here examples from the llama, if you look at the FAB portion, It's actually very small, and it's actually called a VHH single domain antibody. And these are very small. They're about 15 kD, so about a third of the size of a human IgG FAB region. Now, this is a really interesting way of looking at these antibodies. Now, these VHH domains are also called nanobodies. Now, the nanobody or the VHH domain contains a complete antigen binding site, as you can see here. And this is comparing a camelid, so a camel or a llama or an alpaca antibody, again to a human conventional antibody. So if you look just at these diagrams here, you notice one of the stark differences between the nanobodies and the FAB domain of the human antibody is, one, again, they have that single domain, and two, they have this little thing sticking up right here. Now, these three loops represented in these diagrams each represent the CDR regions of the antibodies. Now, the CDR3 loop of nanobodies, or indeed of any heavy chain antibody, is extremely long. So you can see here it's depicted as this much longer chain compared to its own CDR regions and also the CDR regions of the conventional human antibody. Now, this is really critical. So this extended CDR3 loop means that the CDR3 loop of these nanobodies can actually go inside of antigens and see epitopes that antibodies can normally not see. Now, if we look at this a little bit more closely, we can see that it also can, this CDR3 region is also much more flexible relative to human variable heavy and light chains. And this mobility also allows it to move inside and around antigens and epitopes that other antibodies cannot see. And this length of the CDR3 region and the relatively small size of a nanobody compared to the FAB region of human immunoglobulins makes it very, very interesting and has a lot of clinical applications. Now let's go through some properties of nanobodies that make them very interesting molecules to be used not only in the research setting but also in the clinical setting. So compared to FAB regions of antibodies, nanobodies have very similar affinities to their antigens if they're reacting against the same antigen. So that's a really good basis for understanding potentially how antibodies and nanobodies can be used in different ways. As I've mentioned, nanobodies are very small and have this extended CDR3 loop, which means that they can bind to hidden epitopes that aren't accessible to whole nanobodies. But their small size also means that they can permeate better into tissues, and they can also pass the blood-brain barrier more readily than conventional antibodies. And this is a very critical point because you can start thinking about different tissues or potentially malignant tissues that you would want antibodies to go into. Now, also, the fact that nanobodies are single domain is really important, as you don't need supramolecular assembly compared to multi-chained antibodies. So, antibodies, conventional human antibodies, are made up of 4 chains, 2 heavy chains and 2 light chains. So during the assembly of the entire protein complex, you need to make 4 chains of the antibody, and then they need to be assembled together. Having a small protein that's a single domain makes it fairly readily available and makes it much more easy to make in a lab setting. In addition, nanobodies are much more stable compared to a whole antibody. For example, they're much more heat resistant, so they can tolerate much higher and also much lower temperatures compared to antibodies and still remain stable. They're also stable towards detergents and high concentrations of urea. So the fact that they're much more stable molecules and they can tolerate a difference in temperatures makes them very appealing for use in the clinic, as stability, long shelf life, and things of that nature are really important when it comes to drug design. Another thing that nanobodies have that makes them really interesting is that they lack an Fc region. So the Fc region of an antibody is really critical to triggering the complement system, which triggers cytotoxicity of antibody-bound antigens. Now, as you may know, the complement system and the antibody-mediated opsonization system that complement triggers is really critical to clearing pathogens, clearing debris, clearing dead cells from our bodies. However, although these systems are very, very important to keeping us healthy, if we're just talking about developing new drugs or developing new clinical ways of treating a variety of different illnesses or diseases, we may not want to trigger complement all the time. So the fact that nanobodies allow us to tailor the immune response, namely not trigger complement or cytotoxicity, is really important. All these properties of nanobodies means that we can highly manipulate what we target, and by that I mean we can target antigens with a higher efficiency with these extended CDR3 loops, such that we could potentially target epitopes that our normal antibodies can't see. We can also have these nanobodies penetrate into tissues that antibodies can't penetrate into, as well as cross the blood-brain barrier. And also, we're making highly stable molecules for these clinical applications. And we're able to specifically trigger the immune responses that we want. So when it comes to making nanobodies, there's a variety of different ways that this can be done. But today I'm going to be talking about the conventional way that it's traditionally been done. So typically, single-domain antibodies are obtained by immunizing camelids or sharks with the desired antigen and subsequently isolating the mRNA coding for the heavy-chain antibodies. So this is a very similar practice to when you're generating antibodies in the lab, for example, by immunizing rabbits or other animals. So using animals such as camelids or sharks, we can trigger immune responses that they have internally to generate heavy chain antibodies against our antigens of choice. Once this antibody response is generated, mRNA is isolated for heavy chain antibodies, and then we have the specific sequences of these single chain antibodies against the antigen that we would like. Then, phage display libraries can be made to identify clones that bind to the antigen specifically. Again, when you think about the entirety of the population of your B cells in your body, during an immunization, there will be a spike of B cells that are specific for your antigen. However, they're still swimming in a very heterogeneous pool of B cells. And so once mRNA is isolated from B cells in the immunized animals, the mRNA that we get will encode for antibodies that are specific for our antigen of choice, but also for other things. And so we can take the mRNA that we have isolated and use phage display on yeast, for example, to display these different nanobodies in individual yeast cells. Then we can expose these yeast to the antigen specifically that we want, and we can isolate the yeast that grows out, identifying those that are expressing nanobodies that are specific for that antigen, which makes this incredibly powerful. Then, these specific nanobodies that are specific to the antigen can be readily selected for and produced in bacteria. Again, the ability for us to make single-domain nanobodies that are specific for an antigen makes it really easy to generate them in a lab, as they can be made in bacteria. And this ensures unlimited supply that is in a consistent quality. In addition, using phage display libraries, you can take the nanobodies that were generated originally in the antibody, and you can also create nanobodies with point mutations that you personally select for to make higher and higher and higher affinity nanobodies, which makes this an incredibly powerful tool because you can make nanobodies that are specific for your antigen but at varying degrees of affinity. Now, what can be done with nanobodies? Well, there's a variety of different biotechnical, diagnostic, and therapeutic things that you could do with nanobodies. And the applications of this really comes from their small size and unique structure. So let's start off with some biotechnical and diagnostic things you could use with nanobodies. So first, you can use chromobodies. So chromobodies are nanobodies that are fused to a fluorescent protein. So for example, you can have a nanobody GFP fusion. So this is very akin to antibodies that are fused to a fluorescent protein. But nanobodies, because of their small size, can be used to recognize and trace targets in different compartments of living cells it increases the possibilities of live cell microscopy. Now, antibodies are fairly small, especially relative to a large eukaryotic cell. However, nanobodies, again, are significantly smaller relative to the entirety of the antibody. And so these nanobody fluorescent protein fusions are much more readily able to get into a cell and bind to their specific protein target. So for example, if you're trying to understand various cellular processes that cells are going through and you would like to take microscopic images, nanobodies fused to fluorescent proteins, also known as chromobodies, may be a much more highly efficient way of going about this. And again, because we're able to create nanobodies that are have more or less affinity to a specific antigen, you can create nanobodies that are very ideal for microscopy purposes. In addition, another application that nanobodies can be used for is in biosensors. So biosensors are really important in the clinic for sensing the presence or absence of a particular antigen, such as a protein that's associated with cancer. So the way that biosensors work is that you basically have a flat surface onto which you immobilize different proteins such as antibodies or nanobodies. Then you put your patient sample on top of this and look to see how many nanobodies or antibodies have their cognate antigen connected to them. And the small size and high affinity nature of nanobodies makes them really critical in advancing biosensor technology. More specifically, you can put a much more dense population of nanobodies onto a biosensor compared to antibodies. So in this example here, these scientists use an immobilized single-domain antibody, or nanobody, on sensor surfaces to sense human prostate-specific antigen. Human prostate-specific antigen is a protein that is generated most prominently in prostate cancer and is used readily in early cancer development screening. And so you can imagine that if it's early stages of cancer, the concentration of the cancer-specific protein in the blood of patients is going to be fairly low. So we want a highly sensitive biosensor to sense the presence of this protein. Now here you can look to see the sensing, so the ability of the biosensor to sense the presence of the protein compared to the concentration of the human prostate-specific antigen. This line here represents the affinity that VHH has, so the single-domain antibodies have, and these blue closed circles here represent monoclonal antibodies, and these open circles here represent just using the monoclonal antibody FAB region. And you can see that nanobodies highly outperform classical antibodies in this specific biosensor application. However, this has been done for a variety of different other proteins, and nanobodies always outcompete antibodies in this application. In addition, slightly outside of the clinic, nanobodies can be used to aid in protein crystallization. So looking at the structure of proteins at a very, very, very high molecular scale is important in understanding how proteins not only function, but also in the development of drugs. Because if you know the structure of the protein that you would like to target, either by an antibody, or by any other drugs, you can do this much more readily. So nanobodies can function as crystallization chaperones during the process of protein crystallization. And they can also reduce the conformational heterogeneity of the protein that you're trying to crystallize by binding and stabilizing a subset of different conformational states. Now, proteins can move around. A lot of proteins are not static, so a lot of proteins can move and have mobile parts. And the fact that these mobile parts are moving around make them highly difficult to crystallize. The presence of nanobodies reduces the noise and movement that these proteins can make, such that the protein crystal becomes much better. In addition, nanobodies can mask surfaces that interfere with the crystallization process, again optimizing the crystallization process. So those were just a few small examples of the biotechnical and diagnostic ways that nanobodies can be used. But nanobodies have also been used in therapeutic aspects, be that through infections, cancers, blood disorders, really for a high number of diseases that antibodies previously were used for, the nanobodies are being applied to them. So I'm just going to go through a few examples of how nanobodies have been used in the clinic or in preclinical settings.